Hi everyone, welcome to my channel Srinivasa Biology tutorial. For this section to discuss class 11 NCERT chapter cell cycle and cell division part 2. Discuss M phase. To discuss M phase. It is most diagrammatic period of the cell cycle. So it is very important period in that cell cycle. The involving the major the recognition of individually all components of the cell, since the number of the chromosomes are in that parent, the progeny is cell the same. It is also called as an equational division. So here saying about as what? Here one parental cell is present. This parental cell to divide it to forms as two daughter cells. The two daughter cells here they equally to distributed as any the genome or chromosomes equally to distribute as any chromosomes to as any two daughter cells. So it is called as what? It is called as what in a equational division. Why called as equational division? It is equally to distributed. Am I right? So it is called as an equational division. Today, as an convince the mitosis has been divided into as any four stages for of a nucleus division also. That is in a karyokinesis is very essential for understand as in that cell division. In a progressive process, it is very clear cut lines cannot be drawn between the various stages. Karyokinesis involved as in a again four steps. So here the nucleus division is very important for the study of the cell cycle. Okay. Now in this karyokinesis again to divide it as in four steps. The first one is prophase, metaphase, anaphase, and then telophase. How many phases are there? Four phases. Now to discuss one by one. So first to discuss in a prophase. First to discuss as what? That is in a prophase. See, in this prophase, which is the first stage of the karyokinesis of the mitosis, it's following as in S phase and G2 phases. So here the prophase is present between as what? The prophase is present. See here this is in a M phase. Am I right? It is totally interphase like as in a G1 there G1 phase and it is in a S phase, it is in a G2 phase. Am I right? It is in a prophase, metaphase, anaphase, and then telophase. And then telophase. Here they having as in a G0 phase, and here cytokinesis is takes place. So here the prophase is present between as in a G2 phase as well as in what? In a here the prophase is present as in a S phase, and then we are first to discuss here the prophase. The first to discuss as what? Prophase, right? Prophase, which is the first stage of karyokinesis of mitosis, is follows as an S phase and G2 phases. It is present between there. Then it is followed by an S phase and G2 phases of interphase. Now, the S phase, S and G2 phases is a new molecule, new DNA molecules formed are not distinct but interview. The prophase is marked by as an initiation of condensation of chromosomal medium. So in this process, what happened? This prophase, get the totally the chromosome condensation is takes place. Here, the chromosome condensation is takes place. The chromosome material becomes as an the individual during the process of the chromatin, the condensation. So you have to get forms as what? The chromatin, the condensation is takes place here like. This takes place as in a their condensation is takes place. Like the condensation is takes place like. Now after the centrosomes which had the undergo duplication of the S phase of interphase now begins move towards the opposite poles of the cell. So after here forms as what? Two of the and their assets to get formed as into opposite poles when they get complete to get move as in a that completely in that opposite poles. Now the complication, the prophase can be thus marked as in a following characters events. So what kind of characters they are followed, they given is clearly. The first one, the chromosome material condensed to formation of compact mitotic chromosomes. So they completely compact, means mixed, and the chromosomes are seen the composed two chromatids attached to gather in a centromere. So when you see as in a completely one chromosome to get appear as in a light, Now the centromere which add to as an undergo duplication during the interphase. This total the centromere to get duplication takes place in interphase. Okay. Now the begins to move towards the opposite poles. So in this centromere there's present yes now. So here complete to get move from as an opposite poles. Now each centrosome there radiates out the microtubules is called as an asters. So here these are called as what? Asters. These are called as what ma? Asters. Now the two asters together to in a spindle fibers to formation of mitotical apparatus. So here to get forms as a two asters, after they complete to get forms appear like they get forms appear like when it is the end of the prophase. It is in a end of the prophase. Now after here what happens say that cells are end the prophase when the viewed on the under electron microscope do not show as an Golgi complexes, endoplasmic reticulum, and then the nucleus and the nuclear envelopes. See here when we observe as in this total structure, when you see the under electron microscope, the entire the Golgi apparatus, endoplasmic reticulum, nucleus, nuclear envelopes complete does not there appear. Okay, this is in a completely prophase. So condensation is takes place to get forms as in two asters. Okay, okay, this is in a prophase. Next one metaphase. Next one is what? Metaphase. So in this metaphase, the complete the distinction of the nuclear now the metaphase, the complete the distinction of in a nuclear envelope mark the start from the second phase of mitosis. So in this phase what happened, the ended the second after the prophase, the second stage is M phase, in the metaphase, hence the DNA chromosome separated to through the cytoplasm of the cell. So the enter the chromosomes to get completely here separated. Now by this stage what happened? By this stage condensation of chromosomes is complete. In this stage what happened? To complete the condensation of their chromosomes, and they can observe their early the under electron microscope. You see the under closely under electron microscope. This the stage which the morphology of chromosome is most easily to get studied. So how do the chromosomes when they get separated, they get easily to get observed. So that is here the chromosome morphology is seen that the mostly in a metaphase only. You seen that only metaphase. Okay, now this stage is the metaphase. The chromosomes are made up of as a two sister chromatids. So here one observed here the two sister chromatids to get attached on the centromere, to get attached with as a centromere, with or the held together by the centromere. So it is in a centromere now. So in the centromere, the sister chromatids are to get completely they get attached. Okay. The small disc-shaped structure also is surface of the centromere is called as a kinetochore. See here, they having small disc-shaped structure is present. This is called as what? Kinetochore. It is called as what? Kinetochore. Now, this structures several sites of attachment of the spindle fibers of in a chromosome that move to opposite poles of the center cell. So Here the asters as present, these two asters to completely they get forms as in these spindle fibers. To get forms as what? These are their spindle fibers. To get form as what? Spindle fibers. Now, hence this in a metaphase, the character by as in all the chromosomes they're coming alive in equatorial with as in the chromatid of each chromosome connected by as in the kinetochore, the spindle fibers to form as in a one pole, and the sister chromatids is connected by as in a kinetochore to spindle fibers. Then formation of two opposite poles. Here these all chromosomes to get arranged as in a one equatorial plate line, that enter the chromosomes to comes as in a one equatorial plate. This total the chromatids you see that in a equatorial plate of the sister chromatids you see observed only in that metaphase only. Okay, now this plane their alignment of the chromosomes at the in a metaphase, it is reflected as in a metaphase stage. So when these chromosomes they comes as in a that equatorial plates with help of the spindle fibers, this phase is called as what? Metaphase. The key features of the metaphase. What are the important features of the M phase or metaphase? See, in this metaphase, the spindle fibers to attach to the kinetochore of chromosome. So they attach with as the kinetochore, and next chromosomes are moved spindle equatorial and to get their alginate along with metaphase plates through the spindle fibers with two poles. So two poles the chromosomes to get alive with as in a one single plate. Okay, this is what in completely in metaphase. It is what metaphase. Now to discuss anaphase. Now to discuss as what? Anaphase. See in this anaphase, mostly in this anaphase, one of the set of the anaphase, each chromosome to arranged on the metaphase plate, the splits simultaneously and the formation of two daughter chromatids. So here ana means what? Up. Ana means what? Up. So when the two chromosomes that pull up, when they get pull up, what happened? To get form as a two daughter cells. Now they are reflected to as a two daughter cell. The chromosomes of further two daughter nuclei begin to get migration of two opposite poles. So here what happened? It is to get forms as an A, like to get forms as an A, like here two chromosomes to get forms like. Okay, now each of the chromosomes move away from equatorial plate. The centromere of each chromosomes is mainly directed towards opposite poles, hence it leading to it. So these are to get pull up. What happened? That enter the chromosomes to get comes as the edge of the pole. Now within the arms of the chromosomes, they are completely trailing behind. For this the anaphase stage, the characterized by this anaphase characters are mostly using that here. First, the centromeres splits and the chromatids separated. So here they get separated and the chromatids move to opposite poles. Here chromatids also to comes as two opposite poles. This is what in a anaphase. This is what anaphase. Next, after telophase. Next one is what? Telophase. In this telophase, what happened? The beginning, in a final stage of telo means what? End. Telo means what? End. So end phase. In this end phase, what happened? The karyokinesis, the telophase, the chromosomes had reached as the respective poles, their decondensation, lost their individuality. So when the decondensation, the prophase is what happened? Condensation. But here what happened? Decondensation was takes place and the cells they comes as they are again back. Now in this individual chromosomes can be the no longer seen that the set of the chromatid material tends to collect as an two opposite poles. This stage which I show as some features here. So here what happened in this when that completely the cells will be they get completely their decondensation was takes place, the cells comes as that back. Okay, now see the features. The chromosomes, the cluster, opposite spindle poles, their identity loss in the distinct the element. So that entire they lost their characters. Nuclear envelopes develops around the chromosome. So here what happens? Again the nuclear envelopes will be takes place, and the cluster of each pole formation of two daughter nuclei to get formed as a two daughter nucleus. Like now, nucleolus, Golgi complex, and endoplasmic will be there reformed. So that all cell organelles again reform. Okay, this is in a M phase. This is what M phase. Now here what is in a cytokinesis? Here what is cytokinesis? See here in the cytokinesis, the most of the accomplished do not now see cytokinesis. In the cytokinesis, most the mitosis accomplishes not only the segregation duplicate chromosomes into the daughter nuclei, means it does not only the chromosome daughter cells will be not forming, but in the cell it's divided into two daughter cells by the separation of the cytoplasm is called as a cytokinesis, at end of which of the cell division get as a complete. So here the karyokinesis, after when to complete as in a cytokinesis, then only the entire cell cycle is complete. Okay. Now, in which in animal cells, the achieved by as in that appearance of in a farrow of plasma membrane. So, in that animal cells, what happened to get form as in a farrow like? It is to get in a when farrow is takes place, it is to continue to divide it to get forms as in a two daughter cells will be to get forms. Okay, this farrow is gradually that depends and ultimately joins as a center of the divided to the cytoplasm into as an a two. So like it is divided as in a single cells. Now the cells, however, the enclosed by as in the relatively the accessible the cell wall, therefore the undergo cytokinesis in the different mechanism. So the cytoplasm the division is different forms is there in the different organisms. Now see that in plant cells, the wall formation of the start the center of the cell and grow up there outwards to meet as an existing lateral two walls, the formation of new walls beginning the formation of the simple their precursor cell is called as a cell plate. So here also they get formed as in that simple the cell plate will be takes place in the cell plates we get produced as in a two daughter cells. It the already the cell plate is made up of what? The cell plate is made up of as in the middle lamella, that is a calcium pectate and magnesium pectate. Okay, now after at the time of the cytoplasmic division, the organelles like as a mitochondria, plastids get distributed between the in a two daughter cells. The equally that enter the mitochondria and then they are completely the cell organelles will be there equally distributed. In some, the organisms like as a karyokinesis is not followed by as a cytokinesis result formation a multinucleated condition arise leading to formation of syncytism. Example in liquid endosperm or in coconut. So sometimes first they are followed by as what? Karyokinesis. To get followed by as any karyokinesis. Karyokinesis immediately followed by as what? Cytokinesis. Cytokinesis takes place. It is not takes place. What happened? They get formed as any syncytism. Example is what? Coconut water, liquid endosperm. Okay, these different phases. Okay, remaining to discuss in next class. Thank you for watching.